everyone, welcome to your chapter 20 video. I'm making sure I'm recording because I just gave this lecture and was not recording. So we're going to try this again. All right. So in chapter 20, just as a quick reminder, what we are covering are biotechnological techniques that basically use the concepts that we've been talking about all semester to do different things in medicine or different things in science. So this is the application of all of the concepts that we've been going over. In part one, what we talked about was how we can use much of what we've learned about to clone genes. And we clone genes using recombinant DNA technology. So let's write that down, recombinant. And just as a quick reminder, sort of what happened on our last episode, what we can do is we can take a tiny circular piece of viral DNA called a plasmid, okay? We can then use a particular type of enzyme to cut this plasmid. Those enzymes were called restriction enzymes, and I'm going to abbreviate restriction enzymes because this is all review from our last chapter. So these restriction enzymes cut this plasmid open thusly. Okay, so I'm just drawing these little lines right here to show you that this has been cut. We can then take and paste in, if you will, a new piece of DNA from another organism. So in the case from our last lecture, we talked about pasting in the gene for human insulin. At this point, this plasmid is now a piece of recombinant DNA. Because it contains DNA from both a bacteria, or bacterium, I should say, which we have here in purple, and then from a human, which we have here in orange. Our next step is that we can take and we can insert this little piece of DNA, this little piece of DNA into a bacterium, which is what I'm going to draw for you here. So here's our single circular chromosome. We're going to insert our little recombinant plasmid. So there's that part. And we also have this human insulin gene. And what we'll do is we will use the machinery of this bacterium to then transcribe. And I'm going to just abbreviate for transcribe, and then translate that gene and make some human insulin. The other thing is, as this bacteria divides and makes more of itself, what it's really doing is it's making clones of itself. It's making exact copies of itself when it grows and divides. And it's making exact copies of our little plasmid. So in this way, we will have cloned our gene of interest, okay? This process really transformed the field of molecular biology, okay? And we use this all the time in different applications. We talked about genetically modified organisms. We talked about making human insulin. There are many other applications for this technology as well. Today, I want to talk about a process that has been equally transformational to the field of molecular biology. And that process is something you might have heard of before. It's called the polymerase, come on, little guy. Nope. Come on. It's called the polymerase chain reaction. You may have heard of its abbreviation. It's usually abbreviated PCR. And when we talk about the polymerase chain reaction, we typically also talk about a process called gel electrophoresis, which is the process that we use in order to see our PCR results. Okay, so let's jump right in. And thinking about the polymerase chain reaction, or PCR as it is abbreviated. Hopefully you get a clue, don't know why it's doing that. Hopefully you get a clue as to what this process entails by taking a look at this word polymerase. During this course, we have talked about a couple of different types of polymerases. We talked first about DNA polymerase, which can be used to make copies of DNA. We've also talked about RNA polymerase, which we can use to read the DNA and make a copy of it, but the copy is actually in RNA. So what the polymerase chain reaction is going to do is it's actually going to use DNA polymerase to make copies of genes. You can actually think of the polymerase chain reaction as a molecular Xerox or copy machine, okay? What it's going to do is we're going to use it to copy specific sections of DNA, little small sections of DNA. We can actually take a very small sample, like just a little bit of DNA, and we can use this molecular copying mechanism to make a large quantity of DNA. This is how, like, if you're watching one of those police procedural one of those police procedural shows like Criminal Minds or CSI or something like that, and you see a scene where there's just a tiny little droplet of blood, or maybe there's a cigarette butt that's been discarded by the criminal. Well, they can pick up these tiny little samples, which seem like they're really small. We can then use this molecular copying technique to make a large pool of DNA that we can then analyze to try and do things like determine the identity of a criminal. We can also use these large quantities of DNA to diagnose genetic diseases. And so today, what I'm going to talk to you about is how we can use the polymerase chain reaction to diagnose a disease of interest right now. And that is the disease that is caused by the virus SARS-CoV-2. This virus causes the disease I'm sure you've heard of, because this is the virus that causes COVID-19. This is the coronavirus that we are all concerned about. So the name of the virus that causes COVID-19 is SARS-CoV-2, and that stands for severe acute respiratory syndrome. So that's where the SARS part comes in, okay, right here. CoV is just for coronavirus, and 2 is that this is the second coronavirus we found that causes a severe acute respiratory syndrome. The severe acute respiratory syndrome caused by this virus is far more severe than the first one we discovered, okay? So in understanding how we can use the polymerase chain reaction to diagnose whether someone is COVID positive or COVID negative, what we need to do really is understand how this virus infects your cells. So SARS-CoV-2, here is a diagram of it. Let's draw this diagram as well so we can understand the various parts of this virus. This virus starts out with something that looks a little bit like a cell membrane, but it's not a cell membrane, so we're going to give it a different name. Oops, this is meant to be an arrow. We're going to call this the viral envelope. This envelope consists of some phospholipids, like your phospholipid membrane, and embedded in these phospholipids are various different proteins. A protein that is particularly talked about as being embedded in this viral envelope are these spike proteins here, okay? These spike proteins are pretty important to the virus because it's these spike proteins that the virus uses in order to enter your cells. So let's label these as spike proteins. The other thing about these spike proteins is that they are where the coronavirus gets its name. So corona is just Latin for the word crown. Let's look at this nice diagram of a coronavirus. Here, this region here, this is the envelope. And the little brown things are some proteins that are embedded in the envelope called envelope proteins. Here is our spike protein. And if you notice, as you go around, these spike proteins do look a little bit like a crown. So coronaviruses, there's many different coronaviruses, all have spikes on the outside. That's where they get their name. On the inside of these coronaviruses is some genetic material, because we've talked before about how viruses, their whole deal is that they deliver their genetic material to cells. Now notice I've drawn this genetic material as being single-stranded. That is because the genetic material inside of SARS-CoV-2 is actually RNA, okay? The last part of this virus that I want to draw your attention to, I'm actually just going to draw one of them on here. Associated with this RNA are some proteins bound to it, okay? And I'm just going to draw one of them here and I'll color it in. These proteins that are bound to the RNA inside of this virus are called nucleoproteins. Oops, there we go. These nucleoproteins are going to be important in a second because this is the gene that we are going to use in PCR to diagnose whether someone is COVID positive or COVID negative. Nucleoproteins are what are represented in this diagram by all these little green egg-looking things. So they're all just attached to the RNA. So see nucleoprotein and RNA genome. That's what all this is in here. Now, the way that this guy is going to infect your cells, let's draw ourselves a little cell membrane here. I'm going to make it kind of big. So remember, viruses are really small relative to your cells. So I'm going to kind of draw just a segment of a cell membrane. So this right here is a cell membrane. And this is let's just say this is a lung cell. This is one of your lung cells in your body. Now, y'all know from our discussions this semester that your cell membranes have receptors on the surface of them. So here's a receptor right here. We're just going to draw it on here. This particular receptor is actually called the ACE2 receptor. I don't care if you know that or not. If you're interested in what this ACE2 receptor does, that stands for angiotensin cleaving enzyme 2 receptor. Don't worry about it. It has to do with maintaining your blood pressure. But this receptor has sort of a docking area, if you will. Now, in the absence of the virus, this docking area is where hormones bind, and we've seen examples of that before. Once the hormones bind to these receptors, it initiates a cascade of events that changes the cellular functioning. The coronavirus uses the spike proteins to dock with these cellular receptors. And when this coronavirus docks with this cellular receptor, it causes the virus to fuse with the cell membrane and eventually to spit out its RNA genome into your cell. So here we have some viral RNA. Now, what your cell is going to do is it's just going to see this RNA and it's going to be like, cool, cool, cool. We're going to translate this RNA into proteins. So your cell gets tricked into making viral proteins that then get reassembled so that you have a whole bunch of little coronaviruses in this cell. And eventually your cell becomes so full of these that the cell bursts, and those coronaviruses that your cell got tricked into making can go forth and infect other adjacent cells. At this point, I want to stop and show you just a real quick animation of how this works so that we can make sure everyone's on the same page. And then we'll talk about how we can use PCR to diagnose whether you've been infected with this virus or not.
COVID-19 is the short name for the disease known as novel coronavirus disease 2019. Coronaviruses are a large group of similar viruses. Some are known to infect humans, such as SARS-CoV and MERS-CoV. The one that causes COVID-19 is called SARS-CoV-2. All coronaviruses are named for the crown-like spikes that cover their surface, called spike or S proteins. Inside the virus, genetic material called RNA is made up of genes. Genes carry the information to make more copies of the virus. The virus can infect you if it enters your mouth, nose, or lungs. Inside your body, the S protein of the virus locks to a receptor on the surface of one of your cells. This can trigger the virus to enter the cell in a couple of ways. It may cause the virus to fuse with the cell surface, then release its genes into the cell. Or the cell may pull the virus inside by enclosing it in a sac. Once inside, the virus can fuse to the sac and release its genes. Next, the genes use a structure in your cell called a ribosome to make new copies of the virus. The new viruses travel to the surface of the cell. There, they leave to infect more cells. In the meantime, viral S proteins left on the surface of the infected cell can cause it to fuse with nearby healthy cells, forming a giant cell. This may be another way for the virus to spread between cells.
So you can see that the infection mechanism for this virus, as with any other virus, the trick is the virus has to get its genetic material into your cells. So what we're going to do, genetic material, what we're going to do is we're going to use PCR as a way to determine whether that genetic material is in your cells or not. The first step is first to obtain a sample of your cells. And if you haven't had a COVID test, congratulations. If you have had a COVID test, you may know that there are 3 different types of tests. I'm telling you about one of those COVID tests called the PCR test. There's also a rapid COVID test called the antigen test. And there's a 3rd type of test called the antibody test. So we're talking about one specific kind. In this particular test, they take a very long swab. It literally looks like a giant Q-tip. So I'm just going to draw a giant Q-tip here. Okay. And they shove it all the way up your nose until the very, very back of your nasal canal meets the very back of your throat. I'm just going to go ahead and highlight this in yellow. So if you're wondering whether this is uncomfortable, the answer is yes. I have had one of these tests back in the summer. It's not painful. I don't want to discourage anyone from getting the test. It makes your eyes water. It definitely, I wondered if they'd maybe poked my brain. So it's not fun, but the idea is that if they can touch back here, this is an area where if you had inhaled some droplets containing the virus, you might find infected cells. And so hopefully what this little swab is doing is picking up cells from this region. So hopefully we've got, I don't know if you guys can see that yellow, maybe I'll make it a fuchsia color. So hopefully we've got some little cells that we picked up from the back of your throat. Now, what we're gonna do is we're gonna take this swab sample and we're gonna put it in a little test tube. We're going to swirl it around because what we want to do is we want to get these cells that we got off the back of your throat and we want to put them into solution, okay? Then what we're going to do is we're going to take our test tube and let's see, I think I was using this color to make my test tube. And we are going to bust open the cells that we collected from the back of your throat. Now, how we do that, there's a couple ways. You can use a chemical to bust open these cells. So I'm just gonna draw like this. These are busted open cells, okay? So you can use a chemical to bust open these cells. You can use heat to bust open these cells. But the idea is that once you bust open these cells, any of the genetic material inside these cells then floats out into the solution here. So what we're going to do is we're going to use this solution to test whether any of the genetic material in this solution is viral. Now, how are we going to do that? Well, we're going to need some ingredients to make this happen. In order for us to determine whether you have viral genetic material, I'm going to draw myself a big test tube again, because that's how I think about this. I think about making a reaction in my laboratory space. So I'm drawing a ginormous test tube. The ingredients that we're going to need to run this reaction. First of all, we want to make copies of some DNA. So we're going to need a little sample of this genetic material that we collected. So I'm just going to put here, we need a sample that we want to make copies of, okay? And that sample, like I said, comes from this busted open cells and that spit out genetic material, okay? So we're gonna put our sample in here. The next thing, if you remember from talking about how your DNA is replicated inside your cells, you need an enzyme that's capable of copying DNA. And the enzyme that we are going to use is a DNA polymerase, just like we used inside your cells to make copies of DNA. We've been able to isolate DNA polymerase, and we can use it in the laboratory. If we are going to make copies of DNA, we're going to need the basic ingredients of DNA. And if you recall, the basic ingredients of DNA are called DNTPs. DNTP stands for deoxynucleotide triphosphates. Okay, so deoxynucleotide triphosphates. So these are your A's and your C's and your T's and your G's that are going to make up the molecule of DNA. And if you remember from chapter 7, this was way back in chapter 7, these are triphosphates. They have 3 phosphate bonds because each of these A's, C's, T's, and G's that's coupled with 3 phosphate bonds, that's the energy that's going to drive our polymerase reaction, okay? And then the last thing that you need is a primer. Now let's do a little bit of review. If y'all remember when we were copying DNA before, and I'm just going to draw it like this. This is how we drew it before. Boop, 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 boop. Okay, when we were copying DNA before, if you recall, in order to get the process started, we needed a tiny little bit of, we actually used RNA inside your cells. And the reason we needed that is because DNA polymerase, this guy right here, he can't grab onto a single strand. He needs 2 strands to grab onto to get the party started so he can then begin the process of replicating DNA. So we're going to need some primers so that we can get this party started. These primers, first of all, the difference in these primers and the ones inside your cell is that these primers are going to be made of DNA, not RNA. Inside your cells, your cell uses RNA primers. This reaction uses DNA primers. The other thing is we are going to use DNA primers that are specific for our gene of interest, or GOIs. I'm going to put gene of interest. Now, what do I mean by that? Well, in this example, we are interested in amplifying a viral gene. We want to make copies of a virus gene. If the virus is there, we'll make copies. If the virus isn't there, there's nothing for us to copy, and we won't have anything at the end of this process. So what we do, this is a nucleoprotein gene that we sequenced of the virus. So some researcher, when this virus became a problem, went out and determined the gene sequence of this nucleoprotein gene. So that is, to go back, That is the gene that codes for this protein here, okay? We have that gene sequence. What we're going to do is we're going to lay down some primers that are specific for this gene. And it turns out there's actually several different primer sets that we can use. We can amplify this part of the gene, we can amplify this part of the gene, we can amplify this part of the gene. You see where I'm going. This whole thing is the gene. We don't have to copy the whole thing. We can copy this region, or we can copy this region, or this region, or this region. So we're going to put in some primers, and we're going to put in a couple primers just to amplify this area of the gene, okay? So these primers are only going to amplify our COVID genes. They won't amplify anything else, okay? So now that we have our reaction basics, what's going to happen? Well, PCR is going to happen in 3 steps. Let me just get rid of this right here real quick. Okay, so the first step, what we're going to do is we're going to take our reaction that we have in this tube right here, and we are going to put it into a machine called a thermal cycler, okay? And what this machine does, this thermal cycler machine, what it's going to do is it's just going to cycle through a series of temperatures. It's going to go from a hot temperature to a medium temperature, turn the temperature back up, and then it's going to go around and around and around. So in step 1, we are going to turn up the temperature to hot. As a matter of fact, we are going to turn up the temperature to 95 degrees Celsius. Just to give you an idea of how hot this is, water boils at 100 degrees Celsius. So 95 degrees Celsius is pretty hot. What is that going to do? Well, let's just say that in the sample we collected from your throat, we have a viral gene, okay? And I'm drawing it as double-stranded DNA. Now, the viral gene for your SARS-CoV is actually RNA that gets later in the laboratory converted to DNA so that we can use it in this process. So we're going to have some DNA for the nucleoprotein of the virus. Once we turn this temperature up to 95 degrees C, it denatures the DNA. What does that mean? The heat is going to cause these 2 strands to separate from one another. So instead of being together like this, they're going to be open like this. Why is that important? Well, here is why that's important. It's important because in step number 2, we're going to use the fact that these are separated. Let me get rid of this. Oops. Okay. In step 2, what the thermal cycler is going to do is it's going to turn the temperature down, and it's going to turn the temperature down to 55 degrees C. So what that does is it slows down molecular motion just a little bit. So what we have in our reaction are our 2 separate strands of DNA. Boop, 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 boop. Okay. 2 separate strands of DNA. And remember that DNA has a direction. So we'll say that this strand is the 3 to 5 direction strand, and this one goes in the opposite direction. Now, because we've turned the temperature down, our primers, now that everything is slowing down a little bit, our primers that are complementary to regions of this gene can start to line up according to base pairing rules. And you'll notice I put a primer at one end and a primer at the other, because remember, any new strand of DNA gets synthesized in the 5 to 3 prime direction. So here, this one would be 5 to 3 prime. So this new strand is going to get made in this direction, and this new strand is going to get made in this direction, okay? So what happens is at 55 degrees, these primers that are specific for our COVID gene are going to line up, and they are going to combine to these 2 different strands. All right, What happens in step 3? Step 3 is what's called the elongation phase of PCR. And in step 3, what's going to happen is the thermal cycler is going to change temperatures once again to 72 degrees C. So we're kind of at an in-between temperature. Now, why is it going to do that? Well, let's draw what we have so far. We've got our one strand of DNA here, boop, 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 boop. Again, this strand has a direction. Here's our other strand of DNA. Boop, 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 boop. These are meant to be bases all along here. 3, 5. Here's our primer. This is a 5-3 primer. And here's the primer here. Now what we have is a section of double-stranded DNA and a section here of double-stranded DNA. And I've mentioned to you before that DNA polymerase can bind to a double section of DNA. So what's going to happen is at 72 degrees C, that is the optimal temperature for the DNA polymerase to do its job. So it's going to truck along this strand. It's going to line up the DNTPs from our solution, and it's going to create a new strand here. And we're going to have the same thing happen in this direction here. We're going to create a new strand here. So what that means is at the end of this cycle, we will have gone from having one copy of DNA to having 2 copies of that DNA, okay? This takes about, depending on the length of the gene that you're amplifying, this takes about a minute to run this, or a minute and a half, depending on what type of PCR you're doing, okay? Like if I was doing this in the lab, I would maybe do denaturing for 30 seconds, then I would do 30 seconds to give the primers time to find their spots, and then I would give the elongation step about 30 seconds. And I would repeat this, I don't know, 30 to 40 times. I would do this the 30 to 40 cycles. I would go denature, anneal, elongate. Then I would start it again. Denature, anneal, elongate. Every time I run these cycles, I'm increasing my DNA by 2. Check it out. Here is our single starting copy, okay? We're going to denature it into 2, like they have here. And then we're going to let the primers lay down, like they have here. And then we're going to elongate or extend, depending on what you want to say here. So in one round, we had 2 to the 1 copies of DNA, which equals 2. If we undergo this again, so we let these separate and we undergo the process again, you can see that we have 2 to the 2nd, which is 4 copies of DNA. And then if you take this out, here, 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 2 to the 3rd, which is 8 copies of DNA. All right, if you did this over and over, that's not 8 copies of DNA. 4, that should be 16, right? 1, 2, 3, 4, 5, 6, 7, 8. Yeah, that should be 16 copies of DNA. Oof. Bad graphic. I didn't make this, okay? Bad graphic. That should be 16 copies of DNA. All right. Now, if you undergo this process 2 to the 30th, you end up with 10 to the 9th copies of DNA. That is a 1, 2, 3, 4, 5, 6, 7, 8, 9. That is how many copies of DNA that you end up with, okay? So that is a lot of DNA that you get out of just one copy. Now, the reason that we need so much DNA is because the little tiny sample that we started with did not provide us with sufficient material to visualize whether or not you had the viral DNA in your genes. Sorry, to visualize whether or not you had the viral DNA in your cells. We didn't get enough. When we did this, this was not enough DNA for us to see whether or not some of these pieces of genetic material belong to the virus. If we undergo this process, we generate enough sample for us to visualize whether we have enough, like whether or not you have the gene in your sample. If any of this is tripping you up, I have provided you in the notes with a YouTube link that will help you. This was a video that goes over PCR. It's a little longer, so I'm not going to play it here in the context of this lecture video, but it's a really nice summary of how PCR works. Now, the next step is for us to visualize, to look at this DNA. And how are we going to look at this DNA? We're going to look at this DNA using a process called gel electrophoresis. All right. In order to understand this, I want you to visualize a very simple block of basically Jell-O. Think of this as like a Jell-O jiggler situation, okay? So I'm going to try to draw three-dimensional. I'm not the best artist, y'all know that. Okay, so here we have a little block. Okay, this is actually a block of, it's really like a Jell-O jiggler of science, if you will. Instead of being made of gelatin, it is made up of a substance called agarose. Agarose is derived from seaweed, but it essentially, you make it the exact same way. If you've ever made Jell-O, you know you get the gelatin powder and you add boiling water and you stir it up till it dissolves. And then as it cools off, it becomes this gel. Same exact thing. And you can pour that gel into a mold, right? Which is what we've done here. We poured it into a mold that makes it a nice flat brick. But also when we poured it in, we stuck a little comb into it to make these little divots in the block. And these divots are called wells. Okay, now what are we going to put in these divots? Well, in the first well, we are going to put something called a DNA ladder. Now, what is a DNA ladder? Well, it is, again, I'm going to visualize a little test tube for you guys. It is a cocktail of DNA fragments that some of them are big fragments that might be, say, I don't know, 1,000 base pairs long. And you may have a little shorter fragment. Maybe this one's 700 base pairs long. And then maybe this one is 500. Maybe this one is 300. And then maybe you have a little tiny 100 base pair fragment. So a DNA ladder, this is meant to be just sort of a solution. A DNA ladder is a cocktail of DNA that has different fragment sizes, different DNA fragment sizes in it. So we're going to put a little sample of this cocktail into well number 1. In the other wells, we will put our PCR samples. So let's say that we got a sample from Dr. Jones, and maybe we got a sample from, I don't know, some student in our class. Okay. And what we're going to do is we're going to put our PCR samples, these are PCRs that we've already amplified. So we took a sample from Dr. Jones and we amplified it, and now this is our end result, and we're going to put that end result into this well. Now the next step in gel electrophoresis is we are going to run some electricity through this gel. Okay, and we're going to run the electricity through this gel so that we have a negative charge at this end and a positive charge at this end. Now, maybe, hopefully, you remember that DNA has a negative charge to it. So if you're running electricity through this gel, okay, then what's happening is the negative electricity is going to repel the DNA. The positive electricity is going to attract the DNA. And what happens is the DNA will actually travel through the middle of this gel here. So if we were to cross-section it, the DNA is traveling on the interior because on the inside of this gel, turns out if you could zoom in, this gel is made up of kind of like a mesh network of protein fibers that form little pores that the DNA can travel through. So it's going, the negative is going to repel the DNA and it's going to travel through here. And this is going to attract the DNA. It's going to travel through here. The interesting thing is small fragments are able to travel faster because they fit through these little pores more easily. Large fragments are going to travel slower because they have a harder time fitting through these different pores. So you end up with a situation where this gel will sort the DNA by size. So you'll have your large fragments make a band at the top, and you'll have these fragments make a band here, and these fragments will make a band here, and these fragments will make a band down here. So these are your smaller fragments. And up here are your large ones. Okay, now it turns out, so we know, you'll know because you know when you use your ladder, you know what size these different fragments are. So that one is 1,000, that one was 700, that one was 500, that one was 300, and we also had a 100 down here. Okay, turns out that the gene that we are trying to amplify, when we are amplifying the gene for COVID-19, we know that that's about 300 base pairs long. So if I'm positive for COVID-19 and I run this gel and I end up with DNA in this 300 base pair position, that is a positive result. What would a negative result look like? Well, let's just say that I also got, I don't know, Dr. Carter, the president of our university. Let's pretend that we tested him as well. And we're going to pretend that we went through the whole process of PCR and he's PCR negative. And the way we would know that is this would be blank. There would be nothing here. Why would that be? Well, let's go back. If Dr. Carter does not have this gene, then there's nowhere for these guys to line up. So no copying would happen. So when you get here, he would have nothing in this position, okay? Now you may be asking yourself, wait, so we can just see this DNA as you push it through this gel, you can just see it? Well, not exactly. What happens is in addition to putting DNA in these wells, we also put a dye in the wells, and this dye binds to DNA. And it glows under UV light. So here is an actual example of what that looks like in a real gel. This right here, now this is not a gel for a PCR, for COVID-19, this is a different gel, but this is your DNA ladder here. And these are your DNA samples here. And whatever gene this individual was amplifying, this is not my data, this is someone else's I pulled off the internet, but this individual was amplifying a gene that was about 1,100 base pairs long, and you can see that this corresponds to a space in your gene ladder right in between 1000 and 1,200 base pairs. So you have this size right here all sorted out. Just to drive this point home, this is how it works. DNA samples are loaded into the wells, power is turned on, DNA fragments migrate through the gel, with your smallest being at the bottom and your largest being at the top, okay? If you would like more information or you don't understand this, I think, yeah, here I have provided you with a link to a video that explains gel electrophoresis in a little bit more detail. Now, let me just give you my disclaimer. I've actually described for you how you can detect the presence or the absence of COVID-19, okay? It turns out that the way that we actually do this test in laboratories, or if you go to the doctor and get this test, we use a variation of PCR called qPCR. qPCR is just a variation on this technique that not only can tell the presence or absence, but it can tell how much viral DNA you have. And it's just a little bit more complex, so I didn't want to go over it in today's lecture. So this is one application for the polymerase chain reaction. You can use it to diagnose viral diseases. There are other applications that you can use for PCR. You can use it to determine someone's genotype. You can use it in forensic science using something called DNA fingerprinting. Okay, if that interests you, there are videos on the internet that will show you how this process works. And I was going to describe it for you in this lecture, but this lecture is getting a little bit long. So I'm going to tell you a little bit about how we can use PCR for paternity testing. So I'm going to scroll through and I'm going to ignore this. Look up, this is an example of DNA fingerprinting. If you have an interest in forensic science, I highly recommend looking up this process. It kind of tells you a little bit about how you might identify a criminal or a suspect in a criminal case. But I want to talk to you a little bit about how you might use PCR to determine paternity. So it turns out that in human genes, or in your human DNA, we have these regions called STR, which stands for short tandem repeats. They also go by other names. I actually, in my field, we call them microsatellites, and I have that for you down here. They're also sometimes called simple sequence repeats, and all of these terms are very much correct. They're just that different types of scientists tend to use different types of terms. So why is this not writing? Come back to me. Maybe, yeah, okay. So, for example, let's say that individual 1, I think I might have left myself. Okay, so an example would be like, let's just say that I'm going to change out of yellow so it's easier to see. We have individual 1, and in their DNA, they have regular DNA, and then all of a sudden they have these sequences like a common one is GATA. So G-A-T-A, G-A-T-A. So this individual has 2 repeats of this GATA sequence. Well, sometimes your DNA polymerase gets kind of like it hiccups a little bit. If it sees these repeated sequences over and over, sometimes it'll actually accidentally copy them more than once. So you might have another individual that has 3 repeats of this, because the DNA polymerase, when it was copying this section, it accidentally hiccuped and made an extra copy. You might have another individual, so this is individual 1, this is individual 2, this is individual 3. Maybe we have another person who has 4 of these repeats. So you can see that these are short tandem repeats. Tandem means one right after the other. So it's short, it's one right after the other, and they are repeats. Now these repeats are different sizes, aren't they? So because they're different sizes, if we use PCR to lay a primer down here and a primer down here, and we amplify what's in the middle, okay, so we can lay a primer down here, we can lay a primer down here, and we can amplify what's in the middle. We can lay a primer down here, we can lay a primer down here, amplify what's in the middle. Each of these products would be a different size. So if we ran this on a gel electrophoresis gel, these would migrate differently. Right? So let's just draw a little gel electrophoresis gel here. If we were to do PCR on individual 1, their band might be, oh, sorry, their band is smaller. So their band might be down here because it's smaller. So this is individual number 1. If we ran PCR on individual number 2, their band is slightly bigger, so it's going to be a little bit higher up on the gel. If we ran PCR for individual number 3, their band might be even a little higher because this is an even bigger fragment of DNA. Now, what we can do is you inherit these repeats from your mom and your dad. So we can use this to determine whether or not someone is a father. So let's look at an example of that. This is a copy from your book. So here we have mom. We know who mom is because that's the person who birthed the person. So here are some of her bands for some of her short tandem repeats, okay? Because you have many of these. So these are her short tandem repeats. Here's her child. So her child inherited this one from her child inherited this one from her, and her child inherited this one from her. But remember, you get half of your genes from mom and you get half of your genes from dad. So we need to figure out where the child could have inherited this one. Well, if we look at potential father 1, potential father 1 doesn't have a short tandem repeat in this space, but this guy does, right? Okay, so the child has a band right here. He didn't get it from mom. He didn't get it from this guy, but sure enough, he could have gotten it from this guy. The child has a band here. He could only have gotten it from this guy. So what this does is it eliminates this guy from possibility. He could not possibly be this kid's father. Now, this guy could be this kid's father, particularly if the mom knows it's got to be one of these 2 guys, and yeah, this is the dude. But if you were using this for a criminal case, you'd have to be careful because there might be other people in the population that have these genes right here, okay? But you can see how this process would be useful in determining paternity. We can also use it in forensic science as well. All right, hopefully this was an interesting video to you. Remember to use some of those external resources if there's something you don't understand, and I'll talk to you next time about our final biotechnological process that I want you to know from this chapter. Talk to you soon. Bye-bye.